least I have seen him already. Yeah, I started introducing Adrian already. So Adrian did his bachelor in Stanford. Actually, it's interesting. His first paper is, I think, 1998 or so on the depth system. So he was really very early active in the PKS field. Actually, I saw that his second paper, and I must really mention that, is a paper with polyketide. So polyketide is not familiar maybe for everyone here, but it is a really, let's say, NMR breakthrough paper in cell NMR analysis. So polyketide is a full professor in Frankfurt, so I was really excited when I saw that. So finally, so that this also shows how broad the structural biology view is of Adrian on proteins in general. So Adrian then moved for his PhD with the UCSF in San Francisco, and finally, I really would like to mention that this was, I think, as a postdoc with Robert Stroud. He had I think a landmark paper in the field where he characterized the keto-reductase after a limited proteolysis, where he could really start modeling how at least the processing part of a PKS module looks like. I think this was really a great contribution to the field, and it's one of the papers that actually attracted me at least to join the field. And since 2008, he is in Austin, Texas, or University of Texas at Austin, first as an assistant professor, now since 2013 as an associate professor. And Adrian, really thanks a lot for being with us and also contributing even before this talk now already with fantastic questions to this conference.
I'm so glad to be here. So thank you very much, Martin, Helge, for the invitation. And like Jason, I'm so upset that I can't be there with you. I'd love to socialize and chat about our favorite megasyntheses, and we'll just have to do it in the future. But thank you for including me virtually. I have been noticing some messages saying that my internet connection is not so stable, so I hope I don't freeze up. Is the connection good so far?
Yes, it's fine. Yes.
Okay, I'll keep going then. Let me know if something changes. All right, well, I'm going to launch in here with what we've been studying. Of course, I've studied modular polyketide synthases for most of my life. When I was an undergraduate, I was in Keatinge-Clay's lab, and I wanted to understand these modular systems. And I was working at it with the level of making small molecules, feeding it to engineered synthases, and seeing what they made. Once I saw an engineered synthase make an antibiotic derivative, namely an erythromycin derivative, I was hooked. I wanted to keep doing it forever. But unfortunately, this is what we had at the time, just kind of black and white schematics with circles representing enzymes. So I wanted to change that. I wanted to actually see how the chemistry was done, how, find out how nature is doing chemistry, maybe collaborate with it, like everyone in the room, collaborate with nature to make new molecules, hopefully new medicines. How do you do that? How do you figure out the chemistry done by nature? Well, you got to look into that active site that does a lot of the chemistry. And so the tools of crystallography tools of structural biology in general were very important to pick up. So I moved a little bit north to UCSF, and in Bob Stroud's lab, learned some crystallography and started chopping off bits and pieces of the synthases that we love. So we started with the keto-reductase domain and a DH domain, and then an ER came through, and some other domains came through too. We could start to put together a picture, not just my work, but work from many people of what these synthases might look like. Of course, this is still a model, so it's grossly wrong, but we're getting closer to understanding what converts these feedstock chemicals to bioactive molecules that we would love to design. in order to engineer these factories, we have to go back in time to 1990 to get a sense for what it was like when they first sequenced DEBs. So this is the synthase that makes the erythromycin glycone. Everyone's familiar with it. They noticed in 1990 that the second part of the second polypeptide looked an awful lot like the mammalian fatty acid synthase. And that's what led them to coin the term module. They said this bit right here looks like it's repeated, granted not always with the DH or ER or KR, but it's repeated throughout these kinds of synthases. And it seems that that's what we should call the repeating unit then. Starting with KS, ending with ACP, that's what we'll define as the module. But as you want to engineer these synthases, that definition becomes really important. So imagine the car factory where you got the robotic arms and you want to swap the location of the robotic arms that are each doing different tasks, each adding a different bit to the growing product. If you want to do that, you really need to know the beginning and end of that robotic arm. The traditional definition of a module is reinforced when you look at many synthases like the stigmatella PKS that I'm sure Rolf is pretty familiar with. Yeah, if you look on MI-BIG, you'll see in gray kind of the traditional definition of the module, starting with KS, ending with ACP at the N-terminal end and C-terminal end. Of course, at the very N-terminal end and the very C-terminal end, you have docking domains. Here's a class 2 docking domain connecting one polypeptide to another. In fact, this kind of connection, it's an ordered connection, right? And it's got to be relatively strong in order to hold things together so that we can make stigmatella at some rate. So maybe, these non-covalent connections are pretty strong. We can think less about the boundaries of the polypeptide from knowing that. This is an analogy I like to make to my colleagues. You see a bunch of trains. They're not colored. You want to make another train. You want it to be functional. You can grab bits and pieces of other trains that you see out there, but you can't talk to the other engineers. So what do? What bits and pieces do you grab? What's the modular unit of the train? And for 30 years, we've done it like this. Looks pretty, but it usually doesn't work. Kind of makes sense, right? That's the ORF, that's the polypeptide. And you were docking with other polypeptides. But there's an alternate view of a module. And Kira mentioned this earlier. Using this alternate definition, we've been able actually to engineer functional synthases and compare them to synthases that have been engineered with the traditional module definition. We're finding this works better. The trains are working again. I want to tell you about that story. It has a recent kind of start here. This is Lihan Zhang's work, 2017. He and a bunch of other colleagues in Japan looked at these amino polyol synthases. So these are like the mediomycin synthases, right? Make very long amino polyols. And you realize there's 25 to 30 modules right here that are evolutionarily related. Something like this might happen, not just gene duplication or DNA duplication, but transposition of elements, co-migration of domains. You might be able to see that. In fact, they see it right here, and that corresponds to the arrows up here, transposition of a double bond and hydroxyl group within the polyketide product. That's great. That's what we want to do. We want to create new molecules. How do you do this? Well, that's how you should swap things. That's how nature does it. It does it with the KS at the end. KS at the end. And right in the middle right here, it's not colored, but it's the same thing. This stuff that's at the polypeptide junction, that is stable. That migrates together, those domains right there. Lihan was able to get this bioinformatics signal in the sys-AT system. It wasn't easy, but he could see that this KS is most related to that one, so he colored them the same. Same thing with that KS, most related to that one. He did this domain by domain and was able to see which groups of domains co-migrated. And that group of domains, of course, is different than what we term the module. So if you want to engineer a modular synthase, what do you use as your module? This is the way we've been coloring things for a long time, starting with KS, ending with an ACP. I've got black right here, but it really should be colored. Through Lihan and other people's work, we can now color it like this with the KS's at the end. And it makes sense from a chemical perspective too, because some kind of gatekeeping is necessary. When you want to select a double bond over a beta hydroxy, there should be a way to do that. KS should have some kind of gatekeeping system to get that species, which might actually be the minor species on the acyl carrier protein. When you want to do an epimerization, then you have to select for the epimer, the correct epimer. KS has got to do that. Something downstream has got to select for the right species to go on further downstream. This had been seen, of course, with Jörn Peel and others' work, that the KSs and the trans ATs are gatekeeping. I had started looking at the trans ATs back in 2014, hoping I could learn something more about the cis ATs in the process. Of course, they're remarkable in their own right, but I was hoping to get some clues from the cousins of the cis AT PKSs. Lihan John brought outside the polypeptide. This is a depiction again of how you swap around a hydroxyl group and a double bond through recombination that allows you to get that kind of switching on the DNA level, right? It doesn't know anything about the polypeptide junctions, just looking at regions of recombination. You do the double crossover and there you go. You've got a new product at the end and two modules switch places. In the trans ATs, okay, there's no AT within the synthase here. It's relying on this trans discrete protein. In fact, there's other trans enzymes too, of course. The ACPs have got to pick up malonyl units from that AT, but they've also got to have chemistry on the polyketide that they're carrying. So they've got to visit, methyltransferases and ketoreductases. They've also got to visit these kind of enzymes, oxidases, but they're in trans. ACPs have different signatures, actually, which is, again, different than the cis AT systems. I noticed this kind of early on. I said, that's unique. That's different in the trans AT system than is the cis AT system. So we've been studying this for a while. And I'll show you a cladogram in a second that shows how the ACPs are related. But it led us to, again, update the module boundaries in the trans AT system here. So instead of KS through ACP, we've got processing enzymes through KS. The end of one KS to the end of the next KS. End of one KS to the end of next KS. So on and so forth. This is for the nosprin synthase, and the only thing I'm doing differently is coloring them differently by the updated module boundaries. Maybe this is going to help us do evolution or continue nature's evolutionary experiments to move around bits and pieces of the trans AT polyketide synthases. I'll show you on a bioinformatics level what I'm talking about here. If you just grab all the ACPs from the characterized trans AT systems, and there's only 40 or 50 of those, then you do a cladogram. Of course, they're going to group together depending on the context of the acyl carrier protein. This one was kind of noticed early on. Some people in the audience were part of that. There was a tryptophan noticed in these acyl carrier proteins. They're kind of flagging, or the acyl carrier protein has some kind of surface, that's flagging all these other enzymes to work on the polyketide that's there. So that ACP has a different flavor than this one, than that one, than this one. We realized there's hundreds of different module types in the trans ATs, as Eric Hilfrich showed us earlier. But let's just define something like 25 of them. We'll use the letters of the alphabet. We'll say Z is for the others, or uncharacterized. These are pretty well represented. So that's a cladogram we'll work with right there. I can say that, yeah, these ACPs are working with different enzymes, very different enzymes in some cases. All right, I got my ACP cladogram. I've defined the flavors or the module types that these ACPs belong to. What if I do this with the KSs? If I make a KS cladogram, right? Grabbing the KSs that are before an acyl carrier protein. And then color the KS by the flavor of the ACP. This is what I get. Not very much correlation. But if I do this, if I take the KSs that are after an acyl carrier protein, make a cladogram of them, and then color them by the upstream acyl carrier protein, then I get a very nice pattern. So these KSs are very related to one another, even though they're from very different synthases. They are just downstream of an acyl carrier protein that looks very similar to one another as well. So these are co-migrating, and in fact, the whole thing, processing enzymes, acyl carrier protein, KS, they're all co-migrating. We'd started working on these in 2013 or 14. We got a crystal structure from one of the KSs from the basilian pathway. And here's kind of a portion of the basilian pathway shown here. This is a ketosynthase we solved the structure of, and it turns out it's a gatekeeper as well. It relies on a polyketide being processed by an enol reductase before it accepts it. So the polyketide with its beta keto has got to go into keto reductase, dehydratase, and this trans ER right there before it goes on to the ketosynthase. It's a gatekeeper. And actually, we got it with a substrate bound, albeit to the serine mutation of the cysteine that's normally an active site. And the alpha beta bond right here is free to rotate such that it can swing the acyl chain away. If it was an alpha beta double bond, the chain would clash into the tyrosine. We solved structures from other ketosyntheses within the basilian pathway. We got 6 of them. Other workers have characterized ketosyntheses from other synthases. Altogether, we've got about 15 structures now. It might not be necessary to solve too many more KSs because homology modeling is becoming pretty reliable. We're starting to understand, what one ketosynthase looks like by looking at another that's close by it. So if I wanted to solve the structure of this one, well, I just thread through that structure right there. So to get a G, maybe I would just thread through an H, that kind of thing. In fact, the acyl carrier proteins, we got lots of examples of acyl carrier proteins. We can probably homology model through those too. We did that. We were very curious about the ACP-KS connection because it looked like there's a lot of co-migrating residues. And we did this for many KS-ACP pairs. This is just a model that comes out of PlusPro. It's one of the most reliable protein-protein docking programs out there. So with Dima Kozakov as a collaborator, he's the creator of PlusPro, we keep seeing this solution over and over again. There's actually a residue that sticks out. This is a serine residue. It sticks off the surface of the KS. And the cis ATs, it's an asparagine always. In the trans ATs, it can be a methionine or other weird residues that you don't expect to see exposed to the surface. Very interesting complementarity. And of course, it positions the serine, I can't see my cursor right now, there it is. The serine that gets phosphopentadietylated very close to the cysteine, about 11 angstroms away. So it's in position to transfer the acyl group that was just made. If it's the right chemistry, then the KS will accept it. Introducing now the venomizing synthase. Back to the cis ATs. It's just a 2 polypeptide system. And again, this is a model based on crystal structures, but gives us a sense for what this thing looks like. VemG and VemH come together, and then they make venomizing. Very simple, actually grabs a polyketide from a type 3 system, extends it with one malonyl group, another malonyl group, and catalyzes the pyronization. We can look at it from the polypeptide level or the domain level, but for us right now, it becomes interesting to look at it from the module level. If you look at with the traditional module boundaries, this is what you see, right? KS through ACP, KS through ACP. With the updated, we're ending with the KS, ending with the KS, we're ending with the TE. We reconstituted the system, and I'll show you how we did that. Before I show you though, I want to show you just this model in 3 dimensions with first the polypeptide boundaries and the domain boundaries, the old definition of a module, and the updated definition of a module. And it kind of makes sense as you're looking at it to include the docking domains within the modular boundaries. here's the in vitro optimization. We said we got two proteins, we can express and purify them in E. coli. They're phosphopentatenoylated because of the strain we use. If we throw them together with the building blocks, what happens? We get production. Can we optimize it? Let's change the pH. Okay, around 7.5. Change to the salt. 400 millimolar potassium phosphate works pretty well. Reductant, TSEP, 5 millimolar works pretty well. Temperature, around room temperature, because if you heat it up too much more, protein's unstable. Even though it works well, at higher temperatures, it will unfold. And then enzyme concentration. What concentrations do we use? Can we actually measure a KD between the two proteins? We measure something like 5 micromolar. And we get a turnover rate, actually, through the kinetics of 37 molecules per minute per synthase. And it turns out the A domain accepts a derivative, a 3-hydroxybenzoic acid starter unit. So that's nice. Now we're doing polyketide synthesis with modular polyketide synthases in an NMR tube. We just add all the components there. So in water, room temperature, we're watching ATP come down, AMP come up. We're watching the signals from dihydroxybenzoic acid turn into the signals of venomizing in this region here. It's a little noisy, but when we moved to a different system as we scaled up, then things became a little bit clearer. I guess the small molecules are no longer interacting with the proteins because we've got all the proteins here and the small molecules are free to diffuse in and out. Now you can see very clearly the signals from DHBA going down, signals from venomizing going up. Same thing here, mu peak right there because of this, and there's an HD that I'm not showing that's over there. By HPLC, of course, we can watch this as well. So it's green, one pot, inexpensive, semi-preparative, in tens of milligrams, and enzymes can be recycled. We can just grab that bag of enzymes and throw it in a new reactor with fresh molecules. And yeah, we're relying on MAT-B to generate the malonyl and methylmalonyl groups as we go through this study. So let's engineer some synthases. That's what we really wanted to do. Test out Li Han-Zhang and others' hypothesis about the new boundaries. Let's make some hybrid assembly lines. Instead of the first module venomycin, let's grab the first module picromycin. Picromycin is also from Streptomyces venezuelae, where venomyces come from. It's just a different strain. So we put that in there, and sure enough, we get the expected pyrone. So propionyl group starting things off, not that type 3 polyketide. Make the same synthase with the traditional module boundaries. It works too, but much slower. How about the last module? Let's swap that one. This one grabs malonyl groups. That one grabs methylmalonyl groups. Maybe we'll make methylvenomycin. In fact, we do pretty well. If you take the traditional module boundaries, again, there might be this incompatibility, ACPKS, as well as gatekeeping, other things, and we're not doing as well, 0.3 per minute. We don't make methyl pyrone as well as we would like to. That's from swapping out the third and the first modules of the venomycin synthase for the picromycin synthase. It's mainly picromycin modules here, but it works. It works much better, again, than the traditional module boundaries where you've got these incompatibilities. Putting things together on a bar chart looks like this. Wild type working pretty fast, 37 molecules per minute. And these engineered strains, or engineered synthases, working pretty darn well as well with the updated module boundaries. That compared to the traditional module boundaries, it is impressive. You can see the products on a TLC. These are the derivatives. We actually got crystal structures of 5 of the 6 products because we're making 10 milligrams at a time of these molecules. The key points I want you to take home for this part of the talk, and I'm going to get to some ongoing projects in a second, are that module boundaries have really been updated for the cis-AT and the trans-AT polyketide synthases. This is after using the traditional boundaries for a long time. I'm glad that we're starting to try out what nature's been doing as we see it with the aminopolyol synthase that Li Han showed us. KSs often serve as downstream gatekeepers. In fact, Li Han showed us in his supplemental many of the signatures, how you gatekeep for a double bond, how you gatekeep for ER processed polyketide, maybe even for a beta hydroxyl. You can see signatures actually. There's not too many residues at the active site, but there are a few. Just in this one, right after a GVFXG motif, you can see these residues So look for those residues if you're curious, or look at Lehan's web logos of those residues. Silicases are gatekeepers both in the cis and the trans. The updated module boundaries working 100 times in some cases better than the traditional module boundaries. And maybe this is because the traditional module boundaries being used would result in worse contacts between the KS, upstream ACP, or processing enzymes that might be contacting the KS. And of course, you got to worry about the gatekeeping. Maybe the KS that you're putting in there would have the wrong gatekeeping specificities. Venomizing PKS was reconstituted in vitro, making the first assembly on be robustly reconstituted, and a good platform for engineering studies. Reactors were developed, and we're making 10 milligrams of polyketide in half a day. Enzymes can be recycled. Now we're getting combinatorial libraries of polyketides from the in vitro studies, and hopefully one day that allows us to get to bioactive compounds and doing medicinal chemistry with the help of nature. Of course, it'd be great to go from the aromatic compounds to reduced molecules. So let's make some trichoid lactones that have stereocenters. What we do here is we carve out the middle regions of the picromycin synthase. We delete modules. So here we're keeping the first, second, and seventh for making pic 1, 2, 7, and we're keeping 1, 6, and 7 to make pic 1, 6, 7. These have different flavored ketoreductases. If you remember, back to Kira's talk, this one is B2 type. This one is A1 type. So they're going to make enantiomers. These polyketide products are enantiomeric. We can do this in an engineered strain of E. coli that makes methylmalonyl-CoA and get NMR quantities between 10 and 100 milligrams per liter culture of these desired trichoid lactones. You can see the crude extract stained with vanillin right here. Takesh, my postdoc and undergraduate, actually worked on this together and generated these beautiful results. We're working with another laboratory, Synthetic Organic Chemistry Laboratory. Mike Crochet is one of the best polyketide synthesis experts in the world. He's using his own technologies to set some stereocenters, but for him, it's actually difficult to access this molecule, which is a precursor in his synthesis. He can do it, but ends up with very small amounts and it's not very stereo controlled. Those reactions, for whatever reason, aren't generating that ketolactone for him very well. So at this point, we've actually engineered the synthase. We haven't tested it yet, but in the coming week or so, Takesh, Melissa Hirsch, they're going to study this synthase and see if it's making the desired trichoid lactone. So this would be the first case for us for a custom biosynthesis. And hopefully in the future, we're not just making a trichoid lactone. Hopefully we're making dermatolide or something large like that, something with bioactivity. There's a few structural slides. We're looking at these things with cryo-EM. We go to negative stain first and then cryo-EM, and we see big synthases now. We're studying things at the level of the module end of the synthase. This is some people might say it's an iterative synthase, but it's got those docking domains that we've been talking about. So it polymerizes, and you add first a methylmalonyl group, then malonyl group, methylmalonyl group, so on and so forth. We see these structures, these filaments, and we can't wait to get better averages of them to figure out where the domains are located with respect to one another. Here's another synthase. Melissa Hirsch engineered this one. Picromycin, first module, erythromycin, last module. It's a diketide synthase. Not perfect. The ketoreductase doesn't always reduce, and then you get this byproduct that I'm not circling or boxing, but that's pentanone from decarboxylation of the beta-keto acid. But these ones, they're from the diketide acid. You can make lots of that molecule. The enzyme is very easy to purify, and you can look at by EM, and we're starting to get some pictures of that. Of course, the venomizing synthase is one we're going after too. Here's just from the VemG construct. We're seeing some interesting shapes, not exactly like what we modeled, but can't wait to learn how we were wrong. Hopefully we can go after the full venomizing synthase too. And during this strange time, the pandemic time where we're just talking through Zoom conferences, the laboratory united, and a lot of these people are undergraduate, doing a lot of the hard, heavy lifting. What we wanted to do here was understand the loops of polyketide synthases. So we grabbed 1,000 modules and categorized them as alpha, beta, delta, gamma, as you've seen in previous slides. Here's an example of something that's interesting, right? This is one module. We see these repetitive sequences. You've probably seen them too in your synthases. So there's repeats, mysterious repeats, often between domains, sometimes within domains. And you can see even right here on the nucleotide level what's going on. It's actually slip strand mispairing probably that gave rise to this. That's what it looks like at the amino acid level. And it inserted, right? Sometimes between domains, sometimes within domains. We cataloged it all and saw where these things inserted. And you can see with between the domains where it inserts at the updated module boundary between KS and AT. This is actually between KS and the flanking subdomain, or the KS to AT link, or whatever you want to call that bit before the AT. Within the domains, it looks like this, with those red things being loops that these repeats insert into. And I think this is going to be nice for people that want to engineer cut sites or insert domains, fluorescent or modification enzymes. We notice that ACP doesn't have any of these insertions. We notice KS, only 2 examples out of the 1,000 or so that we studied have insertions, and they're both in the same loop. So the KS surface, you can't really modify it. And you'd say, why can't you just get this from the sequence alignment? Well, you can't always tell whether a loop sequence is important or not. Even if it's not conserved, doesn't mean it's not making complementary interactions. But these repeats, you can pretty much guess that they're not important for the structure of the synthase. This is a group of people doing the research. That's Takesh, who featured very prominently. He's been with me for almost 5 years. He might decide to leave for an assistant professorship at some point. I hope not too soon, because he's doing great work. And the graduate students, there's Kate, there's Melissa, and all the rest are undergraduates. And I have to thank the NIH and the Welch Foundation for their support, and all the collaborators and facilities. And thank you for your attention.
So thanks, Adrian, for this very interesting talk. Also, thank you for sharing this new data, unpublished data. So good luck for a structure solution that cryo-EM first images is negative state, and so they look really interesting. So good luck for that. So who has some questions to Adrian? I will start with one. So so my, let's say, personal problem with this, let's call it updated module boundaries, is that this is, I mean, it's really a tough interface. So nature has obviously chosen, like in your train, a very difficult interface, and not the linkers that are maybe unstructured also. So when you design your hybrids, so what is so how do you design module boundaries or the domain boundaries? So, and how often do you actually fail with your construct?
How often do I what?
How often? So you have this venomizing research, the synthesis, the venomizing synthase, and the swap with the trichoid, and so it's a very nice example. So, but I just said, was the hybrid construct you made, was this working with, let's say, the first domain boundaries, the first construct, or how difficult is it to choose the domain boundary between the KS and the AT? This is really a critical interface, looking at the structure.
Yeah, from a structural perspective, you're absolutely right. I know you've studied these KSAT and the intervening domain, whatever we call it, a lot too. That sequence that follows the AT, the LPT, YXF, XXXXXW motif that we all know, that's so conserved. Even though there's small changes here and there, it's so conserved that pretty much all of these modules have it. And that's kind of the glue that's holding together that flanking subdomain and AT to the KS. So you can kind of rely on that, even though it's after where we're choosing our junction site.
So that means you always use the same junction site, and it actually gives high protein quality in most cases.
Most cases, it's worked for us. There was one case, and it was with the venomizing synthase. There's a module 3, I believe. It had a rather long KS to AT linker, and so we made one guess and it didn't work, and we tried another one and it worked. But generally what we do is we go about 10 residues after that GTNAH sequence. at the end of the ketosynthase. So at the end of the ketosynthase, that catalytic domain, it's it's got these conserved residues, and it's not just the GTNAH. There's a few residues after that that are conserved too. And as I think maybe Russell Cox mentioned, some others mentioned, yeah, nature is kind of recombining things on the DNA level where there's a lot of homology, right? So right at the end of the KS, there's a lot of homology right before you go into that linker that we know can be inserted into with these repetitive sequences and such. Sometimes hundreds of residues. Anyhow, that's where we put our boundary, and so far it's been working really well. I think Kira with her constructs is also doing the same at the GTNAH boundary.
Okay, could you take the microphone? Yes. I think it's on. And then I can open it.
Hi, Adrian. It's Kira.
Hi.
I was just wondering the correlations that we see. How much do you think is due to factors beyond substrate specificity? So the AT together with the processing domains determine the substrate that the downstream KS sees. So can that be the explanation, or is it something more complicated? You mentioned that there might be interactions even between the KS and the upstream processing domains in terms of protein-protein interactions, but how would that be compatible with the sort of modular structures that you're proposing?
Yeah, well, gosh, great question. So first of all, I will say there's probably a lot more contacts within these modular polyketide synthases than even we anticipate. When you try to make models, and I'm sure you've done this too, delta modules, they include KR, ER, DH, all those. It's got to be a pretty compact structure because you look at the linkers on both sides of the ACP, they're short, especially the one on the N-terminal end. That linker, and if you realize helix 0 on the ACP, but you're sometimes dealing with just 5 residues or 10 residues. It's a short linker. How does ACP get everywhere? Well, actually, we believe that the last part of the ketoreductase has a chance to unfold a little bit, maybe even through a KS-KR interaction. Maybe that KR interacts with the KS loops, which are conserved, and you free up those residues so the ACP has a little bit more mobility. Anyhow, there's connections with the KS, downstream and upstream, and we're seeing lots of connections. My group is doing this PKS puzzle project right now, trying to fit all these solved structures together. And that's what you realize. There's a lot of contact. And so, in some senses, solved structure of like a KSAT didomain, you're like, well, that belongs together structurally, right? Or other things that we could solve the structure. There's going to be a lot of contacts, both upstream and downstream of the KS. And so when we do these swaps, yeah, we're thinking about those contacts. Let's maintain the natural contacts between those upstream processing enzymes that maybe are contacting the KS downstream and the ACP KS contacts that we keep talking about. You mentioned also the gatekeeping, how much to think about substrate specificity related maybe to those contacts. I think it's maybe equally important. I don't know. We don't have the numbers, right? But I think probably those protein-protein interactions are very important, and the gatekeeping interactions in many cases are very important too. Of course, it is dependent on the exact module type that we're talking about, the exact polyketide we're talking about. But in general, I think there's both gatekeeping and those interactions you need to watch out for when you're creating a new junction, if you're using the traditional boundaries.
So anyone with another question? Otherwise, I would say we move ahead. So thanks, Adrian, for this very exciting talk, and I hope you will stay in this meeting still for. So just if you have other questions, questions, other talks, just ask them. Otherwise, let's thank Adrian again, and let's